All right, welcome back. Today we're going to put on our detective hats and dive into a real neurological mystery. We're going to follow the clues, starting with just one tiny faulty gene, and see how it leads to a total breakdown in the brain's entire communication system. It's a wild ride. So it really all boils down to this one question. A shaking rat and a complex human disease like multiple sclerosis, they seem like they're from different planets, right? But believe it or not, this little animal is holding a massive key to unlocking the secrets of what's really going on at a cellular level in some of these devastating diseases. Okay, first things first, let's meet our subject. This is the Long Evans shaker rat, or let's just call it the LES rat. Now, these guys, starting when they're just 2 weeks old, get these really severe tremors and have a super hard time with movement. Scientists figured out pretty quickly that it was all because of a single genetic mutation, but the big question was how? How could one little mistake in the code cause the whole system to fall apart? And that, right there, became the mission. You gotta think about it like you're a detective at a crime scene. You see the end result, in this case, the tremors, but your job is to work backwards, to follow every single clue and figure out exactly what went wrong and why. So, after digging around a bit, our scientific detectives started to close in on a prime suspect. And that suspect is a material that is just absolutely crucial for your nervous system to work properly. It's called myelin. So, what even is myelin? Well, the best way to picture it is to think about an electrical cord. You know that plastic coating on the outside? That's basically what myelin does for our nerves. It's this fatty layer that wraps around our nerve fibers, our axons, and it keeps the electrical signal contained so it can zip along super fast and efficiently, getting the message exactly where it needs to go. Now, here's the problem for the LES rat. That genetic flaw, it messes up a single protein, something called myelin basic protein, or MBP. And without that one specific protein, the myelin sheath just can't wrap itself tightly. So instead of a nice, snug insulation, you get this loose, floppy covering, or sometimes nothing at all. The wire's basically left completely exposed. Okay, so we've got our suspect, faulty myelin. Now the investigation moves to the actual crime scene, which in this case is the cerebellum. That's the part of the brain that handles motor control. And the researchers zoomed in on these really big, important nerve cells called Purkinje cells to see just how bad the damage was. And wow, this chart really tells the story. What you're looking at is something called the G ratio. It's just a fancy way of measuring how good the myelin insulation is. Here's the key. A higher number is bad. It means the insulation is thin or non-existent. So, you see the healthy rat on the left? Low score, nice thick myelin. Now look all the way to the right at our LES rat. That score is off the charts. It's proof positive. Its insulation is a total wreck. But it gets worse. It wasn't just a case of missing insulation. The actual nerve fiber, the wire itself, was starting to fall apart. The researchers saw these weird swellings popping up along the nerve, and the special parts of the nerve that help the signal jump and move fast, they were all messed up. Even scarier, the very tips of the nerve, the parts that actually send the message, were just degenerating. So the wire wasn't just bare, it was literally fraying at the ends. So you see all this physical damage. And the obvious next question is, right? What happens when you try to send a message down this broken, frayed wire? Well, the answer is pretty mind-blowing. 100%. That's the failure rate. Every single time the researchers tried to get these damaged nerves to send a signal, it failed. Not sometimes, not most of the time, every single time. So let that sink in. The communication line was completely, totally cut. The message would leave the first nerve cell, the Purkinje cell, and then nothing. It just never arrived at the next neuron. It's like picking up the phone and the line is just dead. Total silence. But here's where our detective story gets really, really weird. Think about it. If the neuron sending the message is silent, the neuron that's supposed to receive the message should be quiet too, shouldn't it? Well, when scientists actually checked on those receiving neurons, they found the exact opposite was happening. Take a look at this. This shows you how often the receiving cells, these are called DCN neurons, are firing. In a normal healthy rat, they fire maybe 2-3 times a second. But look at the LES rat. They're going wild, firing more than 7 times a second. They're totally hyper. So that's the paradox, right? How on earth is the receiving end going crazy with activity when the sending end isn't saying a thing? And here's the solution. It's so clever. It turns out those Purkinje cells, the ones that went silent, they don't send go signals. They send stop signals. They're inhibitory. Their entire job is to tell the next neuron in line, that DCN neuron, to chill out and slow down. But because the myelin is busted, that stop signal never gets there. It's exactly like someone cut the brake lines on your car. Without anything telling it to slow down, the DCN neuron just goes nuts, firing completely out of control. Okay, so let's tie this all together. Let's look at all the evidence and deliver the final verdict in this case. You're about to see just how critical that tiny little fatty sheath around a nerve really is. This chart really lays out the whole chain reaction. It all kicks off with one tiny genetic mistake. That mistake means you get no proper myelin. Because there's no myelin, the nerve fiber itself starts to fall apart. And that means the crucial stop signals never get delivered. As a result, the receiving neurons just fire like crazy, which creates total chaos in the brain's motor control center. And what's the end result that we can actually see? Exactly what we started with, the tremors and the motor problems in the rat. It's a perfect, tragic domino effect. And the conclusion from the researchers themselves just nails it. The real problem here wasn't just a slow signal. No, it was a total failure of the brain's braking system. This shows us that myelin is so much more than just insulation on a wire. It's a critical piece of the machinery that makes sure those stop signals get where they need to go. And here it is, the big connection. This whole domino effect we just walked through. The myelin disappearing, the nerve fiber breaking down, the stop signals failing. This is an almost perfect mirror of what scientists see happening inside the brains of people with multiple sclerosis. So this little rat gives us an incredible window, a way to understand the nitty-gritty mechanics behind symptoms like tremors and coordination loss in people. So that leaves us with one last big question to think about. This entire neurological catastrophe, it all started because of one single missing protein. It just makes you wonder, right? What other tiny essential pieces are out there holding our entire nervous system together? And how many more of these incredible mysteries are just waiting for the next team of scientific detectives to come along and solve them? Thanks for digging into this with us.